Hi, I'm Jessica Bruhn. I am the microED group leader at Nano Imaging Services, and I'm here to tell you about our small molecule microED structure determination pipeline that we've built for the pharmaceutical industry. So microcrystal electron diffraction, or microED, is part of this umbrella of 3D electron diffraction techniques. This technique works by placing crystals onto a transmission electron microscopy grid, exposing these crystals to an electron beam, and then recording a diffraction pattern on the camera below. When this is coupled with continuous rotation of the stage, we collect diffraction data in a manner that is analogous to the way that most X-ray crystallography data is currently collected, which gives us the great advantage of being able to use X-ray crystallography programs to process our data and leverage the many, many decades of advancements in the field of X-ray crystallography. So you might be wondering why I am speaking in the cool structure session at the ACA meeting and not the electron diffraction session that was on Sunday. And I want to assure you that this choice was intentional and that I'm just really, really excited to talk to this group specifically today, because I really feel that small molecules are where we are currently seeing the greatest benefit from electron diffraction and microED specifically. So here's a look at the number of unique depositions into the protein data bank and the Cambridge structural database for proteins, peptides, and small molecules. And as you can see, there in the recent years, there's been a huge increase in the number of small molecule and peptide structures that have been deposited, and really highlights the rapid growth of this technique for these types of samples. So you may be wondering why it is that there's been this more rapid growth for small molecules and proteins. And to understand that, I think it's important to understand some of the differences between these types of samples. So first is that the crystals for small molecules are typically, or at least the ones that we're working with, are often supplied as these dry powdered crystals, which we can very easily place onto a grid and put into the microscope and collect data from. Whereas protein crystals, on the other hand, these crystals are typically grown in aqueous solutions, and the crystals themselves are actually composed of about 50% solvent by volume. And so these protein crystals generally get quite unhappy when you try to take away that buffer that's surrounding them, and they will lose their ability to diffract well. And so kind of managing that need to keep some buffer around the crystals without drying them out too much can be quite a challenge for grid preparation for protein microED. Additionally, microED is very sensitive to the thickness of the sample, and if additional thickness in the form of buffer above and below the protein crystals, this can cause a lot more challenges when preparing grids. And a lot of groups have turned to focused ion beam milling of these crystals to produce thin lamella for data collection, but that adds another very expensive instrument and additional expertise that's required to get this up and running in your own lab. So that's one reason that small molecules have been, they're easier to work with and have taken off a bit faster. The other is that small molecule crystals are composed of many very, very tiny unit cells. So for the same volume of crystal, you have many more copies of the unit cell. And that is additive in the intensities of the diffraction spots that you see. So the more unit cells you're exposing with the beam, the brighter the spots are going to be. Proteins, on the other hand, they have pretty large unit cells. And so that there are fewer unit cells being exposed for the same volume, and that leads to lower intensities. And this can be somewhat overcome by increasing the dose that you're applying, but that can lead to radiation damage. Additionally, small molecules generally diffract to really high resolution, and there are many robust tools for phasing small molecule data. And so all these things together just really make it such that small molecules are seeing the greatest benefit from electron diffraction today. So as most people in this session should know, crystal structures are of small molecules in particular of huge importance to the pharmaceutical industry. They provide a very, very high confidence model in the compound you're interested in, far more so than NMR or mass spectrometry. Additionally, relative configuration of all stereocenters is possible through crystallographic techniques, and often absolute configuration, so understanding the chirality of every single stereocenter within your molecule, is possible using crystallography. Additionally, understanding the crystal lattice that is forming these crystals is of great importance for those working in the formulation space, as the lattice properties greatly impact things like solubility and stability, leading to differences in pharmacokinetics, which are very important for the pharmaceutical industry. And I'm sure many in this audience are also aware of this paper that came out in 2014 from Kim Janda's group at Scripps, where they went to synthesize a compound that had been patented and was about to enter clinical trials, and found that the compound they synthesized wasn't active. So they took that, they sent that to for single crystal X-ray diffraction studies, and actually found that the active compound was not the same structure that had been patented in the initial patent application. And when looking at these molecules, you would see that it's going to be very difficult to distinguish these by mass spec and NMR. And so really it's crystallography that is providing you this really high confidence in your connectivity of your molecule of interest. All right, so let's kind of compare some of the common crystallographic techniques used for small molecule structure determination. So by and large, single crystal X-ray diffraction is the most commonly used technique. It provides really, really high quality data, quick turnaround times once you have a crystal. It's a very well-established technique, and using modern collection techniques and data processing, you can usually get absolute configuration out of this data. This is definitely the gold standard and the method of choice for people working in small molecules. The big cons here, though, are that it does require a suitably sized crystal, which can be challenging for some samples. And so here we kind of run into size limitations. These numbers are, of course, debatable, but for a home source diffractometer, you might need crystals about 50 microns or larger. Most small molecule structures are determined on home sources. If you're a lucky group and you're able to get synchrotron time for your small molecules, you can maybe get away with slightly smaller crystals. But really, there is a fine limit at which point you can no longer collect data. And this is where microED really shines. So microED is able to collect data from crystals down in the 0.2 to 2 micron range, which greatly expands the number of crystals that we're able to determine by crystallographic methods. And another big advantage of microED, especially in comparison to other related electron diffraction techniques, is that because we're collecting data using that continuous rotation method, we can process this data in software that was written for processing X-ray crystallography data, and just, again, leverage those decades and decades of hard work from programmers in optimizing this process. Additionally, we can always get relative configuration information out of a crystal structure. So if you know one stereocenter in your molecule, you can infer the chirality of the other. Absolute configuration is a bit more of there's a lot of interest and movement in that. And if you're interested in understanding where the field is there, I encourage you to read this paper by the Platanis group in Science 2019. Additionally, microED does have the ability to localize hydrogen density a bit better than X-rays. I have this in gray because I do feel that this is kind of resolution dependent. So if you have lower resolution data, it will be harder to see these hydrogen atoms. But again, because electrons interact with both the nuclei and the electron cloud, they are better able to see these hydrogens than X-rays. The big cons here, though, are that this is an emerging technology, so things are changing pretty rapidly. There's a very high cost due to expensive equipment. So at Nanoimaging Services, we're using a Glacios microscope equipped with a Zeta D camera. That microscope's about $1 million, and the camera's another $500,000. So that very expensive instrument leads to higher costs relative to benchtop diffractometers. And then the data processing it poses some unique challenges that most small molecule crystallographers are not used to, such as combining data from multiple crystals and dealing with radiation damage. I'll also mention and kind of put a comparison in with X-ray powder structure determination from X-ray powder diffraction using bright field refinement. So this is another technique that has a very nice size range. There's no real lower limit on the size of crystals accessible to this technique. But the big cons here are that to get high enough quality data to actually solve your structure from it, you probably should be using synchrotron radiation, which not all groups have access to. And it can be very challenging to determine structures from such data. There's a lot of peak overlaps, kind of depending on the size of your unit cell, and high resolution data can be hard to pick out. And so you have kind of this higher propensity of the model coming out of here potentially being incorrect. So in that regard, I would say microED is a better choice when you have small crystals than doing right field refinement. All right, so here's a look at how the microED pipeline at Nanoimaging Services works. So we're asking our clients to submit samples as dry powdered crystals. We're asking that they send about 1 milligram of sample. We have ways of working with less than that, but that's kind of a comfortable amount for us to scrape out of a vial and get onto a grid. We also are asking our clients that if they're able to, that they collect X-ray powder diffraction just from a benchtop diffractometer, just to confirm that their sample is actually crystalline and worth spending the time and energy of putting it into the expensive microscope. And we found that this is a really great screening tool and making sure that we don't end up putting amorphous samples into the microscope. So for grid preparation, it's pretty easy. We just take this little TM grid, it's about 2 millimeters in size, and we'll just kind of press that up against the powdered sample, and crystals will just kind of stick on there through static charges. And we can cool that down to liquid nitrogen temperatures and then place that in the microscope. So from there, we will use the microscope in imaging mode to find crystals, and then we will put the microscope in diffraction mode to collect data sets. And so for easy cases, we'll typically collect about 20 data sets, and that between finding the crystals and collecting all of that data, that takes about 2 hours of time in the microscope. For more challenging cases, we might collect somewhere between 50 and 150 data sets and take 4 to 8 hours in the microscope to do that. Occasionally, if we put the first grid in the microscope and see some problems there, we might go back and prepare another grid, potentially adding a heating step to remove water or low molecular weight contaminants, or we might try resuspending the crystals or charging the grid to kind of get better crystal density onto the grid. Once we have our data, we'll move on to data processing, which is of course broken into the data reduction component and the phasing component. And so at Nanoimaging, we are processing our data with the program Dials, which allows us, we individually process each data set, and then Dials has some nice tools for doing scaling of multiple data sets together and analyzing which are your better data sets. And if you're interested in learning more about this, you should check out David Waterman's talk that was given earlier at the ACA meeting. And then once we have our reduced data, we will attempt phasing. So typically we'll start off trying to phase with ShellXT. If that doesn't work, we'll move on to ShellXD. And then if we're really desperate, we'll try molecular replacement in phaser. This is a very iterative process, and we often go back and try different combinations of data sets, different indexing strategies, different scaling procedures, and we'll just keep doing this until we're able to phase the data. On occasion, we'll also go back and collect additional data if we're just really struggling to solve it with the data that we've currently collected. Crystals can be very heterogeneous in how well they diffract, and sometimes if you just keep shooting enough crystals, you'll find one that diffracts to much higher resolution than the others. And these grids are covered in thousands and thousands of crystals, so it's not hard to keep collecting more data when you're struggling. So at Nanoimaging Services, we're collecting data with the program Leginon. And so in 2019, Anchi Chang, with some help from me, added in the capability of this program to collect microED data. Leginon is also the program we use to collect all of the data that we collect at Nanoimaging Services, so single particle cryo-EM data, images of liposomes. But yeah, in 2019, Anchi adapted it to collect microED data. So this works by, first, we'll take low magnification images of the grid, and this allows us to look for crystals. Once we find ones that we like, we can take preview images of these crystals in diffraction mode and see what kind of diffraction we're getting from a particular crystal. If we like the crystal, we can add it to our queue and submit it for automated data collection. Automated collection works by first centering the crystal meticulously in X, Y, and Z. Our beam is smaller than the crystal, so it's very important that we center the crystal as best we can with the limitations of our goniometer. So next, the Leginon will turn the microscope into diffraction mode. We're using about a 600 nanometer size beam that will expose the crystal with. And we will couple this with continuous rotation of the stage. So we're collecting data typically from -60 to +60. And this is about what the limitation of the stage is. We're not able to collect beyond -70 to +70, which can lead to slightly lower completeness data from single crystals. And this is, again, it's coupled, continuous rotation coupled with continuous collection of the data with the camera in rolling shutter mode. So we minimize data loss in between frames. So we recently published a paper where we talked about our experiences looking at the first 50 or so samples that we taken in. And I encourage you to check out that paper, but I'm going to summarize those results in this talk. And so I first want to start off by telling you about the types of samples that we've been looking at. So for the client submitted samples, we're looking at samples in the size range of about 400 Daltons to up to 1,000 Daltons, which is really encouraging to see that we are solving structures of these rather large compounds, which are kind of getting away from Lipinski's rule of 5, which is great to see. Additionally, for the samples that we knew the atom types involved, we know that a lot of them did not contain any heavier atoms, so things like sulfur. And so we were really excited that we were still able to phase data when maybe carbon or oxygen is the heaviest atom in the whole molecule. So that was really exciting for us. Here's a look at the outcomes, typical outcomes. So of the first 60 compounds that we looked at, 3 that were submitted to us, we were never able to find crystals. This was kind of before we started asking clients to do powder diffraction before sending us samples. And I think that powder diffraction would have prevented us from putting those samples in the microscope in the first place. And then in 6 of these 60 samples, we ran into a situation where we weren't able to get enough data to solve the structure, or there were some other problems with data collection, like water on the grid. And so we were saying that data from those 6 samples probably could not be solved. But we do estimate that the other 51 out of 60, we did collect enough data that we do think these data were solvable. Some of those structures were solved internally and many were solved externally by our clients. So talking about resolutions that we achieved. So I've plotted here the resolution of all of the data that we collected that we deemed processable. So in most cases, we were achieving resolutions of 1.1 angstroms or better, which is great to see, especially given that that's kind of the line where phasing gets much more challenging. We were still able to phase data for some of these lower resolution structures, including a 1.6 angstrom structure, but things do get more challenging there. All right, so another common concern people have with electron diffraction data is what about completeness, especially because of the limitations of our goniometer. So here's a look at the plot of the histogram of the completenesses of various data sets for all the data that we processed. And in about half of the cases, we are able to collect enough data from enough crystals that when we combine them together, we'll get 100% completeness. But there are some cases where we are getting lower completeness data. Most are falling in the 90% range, but some are down in the 80s. I will say that this has not prevented us from being able to solve these structures. And we, in the paper, include one structure from a data set that's only 84% complete. All right. So here's a look at how we are phasing these. So for the ones that we phased internally, about half the samples could be phased with shell XT, which was exciting because that's kind of the method of choice. It's very easy and you don't have to necessarily have your space group right at the beginning, so that's great. For more challenging structures, we would turn to shell XT. For even more challenging structures, we would turn to phaser and molecular replacement, or maybe a combination of phaser and shell XT. Here's a look at the refinement statistics that we were getting out of all the structures that we've fully refined. And you'll note that these R values, they are higher than what we would expect for single crystal X-ray diffraction, but they are pretty good for electron diffraction data. And I'll just note that these data were refined using kinematical refinement, and we did not account for dynamical scattering. And presumably, if we were to refine that dynamical scattering, these R values would drop. I'll also just note that we did not refine the extinction parameter. So one last thing that I want to kind of leave you with thinking about is the relationship of a microED structure and powder diffraction, actually. So one kind of great advantage of all these techniques, all these related crystallographic techniques, is that you can take a structure determined by microED, say this one here for progesterone, and you can simulate an X-ray powder diffraction using the program Mercury here. And you can compare that to experimentally measured patterns. And this is just a really valuable tool for chemists who are in the lab trying to figure out if they have what they think they have, and kind of, they can always compare an experimentally measured pattern with what they would expect based on a microED structure. And this is just a really valuable tool that a lot of our clients have enjoyed using. All right, with that, I'll summarize and just say that microED structure determination is much easier for small molecules compared to proteins, which is likely contributing to its faster rise in adoption, both in academia and in industry. I'll also just state that microED is a wonderful complement to single crystal X-ray diffraction when a high quality model is required and crystal size is limited, or maybe there's just not enough time or material for crystallization trials to get a crystal suitable for single crystal X-ray studies. All right, so at Nanoimaging Services, the typical molecules submitted by our clients within the pharmaceutical industry ranged in typical resolutions achieved were about 0.9 to 1.1 angstroms, with a few in the 1.1 to 1.6 angstrom range. And additionally, about half of our samples analyzed achieved 100% completeness when we combine crystals from data from multiple crystals. And we really expect microED adoption to continue to grow within the pharmaceutical industry, with contract research organizations such as ours, Nanoimaging Services, playing an important role in kind of driving that adoption. With that, I will leave you with my acknowledgement slides. I really want to thank Clint Potter and Bridget Carragher, the founders of this company, for giving me the opportunity to kind of pursue this new endeavor that the company is embarked on. I want to thank Angie Chang for all of her help. None of this would be possible if it weren't for Angie configuring Leginon to collect really, really high quality data. Giovanna has been really helpful with processing data and thinking about crystallography in meaningful ways. Melanie also was helpful in writing our grant. Brandon Reed was helpful in processing all of the data for the paper and writing some scripts to automate things. Brandon Mercado at Yale has served as a consultant for us and has taught me, a protein crystallographer, everything that I know about refining small molecule data, and he's just been great to work with. David Waterman at CCB4 has been incredibly helpful in getting us up to speed on how to process electron diffraction data in dials and thinking about better ways of handling this data. That's been really helpful. And then I just want to really thank the microscopy team and our IT team for all of their hard work collecting the data, getting all of our systems in place to do this. And then especially I want to thank our clients for kind of learning with us and sharing as we've been figuring this out together. Also want to thank helpful conversations with people in the field, crystallography field and microscopy field, and just especially Tim Grun. I've had a lot of really, really great conversations with him about electron diffraction and then our grant. So with that, I will stop and let me know if there are questions. Thank you.